All right, so opposite charges are attracted to each other. Energy is required to separate the charges. Energy is liberated when the charges come together. And if we have a difference on one side, or like if you have a membrane and you separate your charges across that membrane, then it gives us what we call potential energy. So the separation of a charge causes potential energy. Okay, so some definitions to think of when we're thinking about electricity. First, we have what's called voltage, and this is a measure of this potential energy across the membrane. We measure it generally in millivolts. So our voltage is a measure of the potential energy that we have across the membrane, and it's expressed in millivolts. The next definition that we have is current. Current is the flow of a charge from one area to another. So in the case of the body, it's the flow of ions between 2 points, and this current can be used to do work. So this is the flow of charges. In our case, in the body, these are ions between 2 points. So then the last definition is resistance. And resistance is like a hindrance to the flow of our current. So it's anything that's going to hinder or slow down the flow of our charge. So in the case of like an action potential going down the axon, the resistance is all of the organelles that you may find in that axon, or proteins, or the neuroplasm, the fluid in there. That's all the resistance in the axon. An insulator is also giving resistance. So there's resistance to the flow of charge out of a myelinated axon because it has it's got that insulating effect. So we could say that an insulator has a high resistance. And so our myelin sheaths are acting as insulators because they give a lot of resistance to losing that charge from the axon. We'll talk more about our different types of channels that are going to allow our cells to behave the way they do. So we've got different types of membrane channels that we'll start with our general definitions of those, and then we'll look at how they affect our cells and affect the conductivity of our cells. So ion channels allow things to move down their concentration gradient. So our channels will allow ions to move down the concentration gradient. Pumps move things up the concentration gradient, and so they require energy, so they use ATP. There's only one pump, well, there are 2 pumps that we're going to talk about, so we'll just kind of give a general definition for now. Our pumps are actively pump ions against their concentration gradient. So the first type of channel that's going to have an effect in our neurons are what are called leakage channels. And leakage channels are just always open and allow an ion to go down its concentration gradient. So I'm going to draw a general cell over here. We'll put in our different types of channels. So our first types are leakage channels. So we'll call this purple one a sodium leak channel. And it's always going to be open and allow sodium to leak into the cell. This orange one we'll call a potassium leak channel. And it's always going to allow potassium to leak out of the cell. The next types of channels we have are what are called gated channels. And gated channels, it's like a door that's shut and locked. You need a key to open that lock. So to open a gated channel, we have to have some kind of key to open that lock. We've got different types of gated channels. So for gated channels in general, we'll say they require something specific to open. And this specificity is important. So the first type of gated channel that we have, we've already talked about when we talk about the neuromuscular junction. The first type of gated channel are chemically gated channels. And they require a specific chemical or neurotransmitter to bind to open them. So the key that opens the lock is a specific neurotransmitter. So chemically gated require the binding of a specific chemical. And in this case, it's a neurotransmitter. And this specificity is really important. So we've got lots of different types of neurotransmitters. Well, not lots of different types, but we have different types of neurotransmitters. And you can't use one to open a specific chemical gate. So an acetylcholine gated channel will only be opened by acetylcholine. A dopamine channel will only be opened by dopamine. Receptor ligand specificity is very important in both the nervous system and the endocrine systems. You have to have the exact right chemical or the channel won't open. So chemically gated are our first type of gated channel. The next type of channel we already also met is the voltage gated channel. And this is going to require a specific voltage. So, and again, a voltage is a difference in charge that we can measure from one side to the other. So it's going to require a specific voltage to open. Then the last type of gated channel that we're not going to talk about right now with neurons, but we will talk about when we get to receptors, is called mechanically gated. And it requires some specific type of physical perturbation. So like, there's a difference between the receptors that feel light touch, and the receptors that feel deep touch, and the receptors that feel pain, and the receptors that feel vibration. So all of those are mechanically gated, and they are opening in response to a specific type of physical perturbation. So how do our gated channels work? This figure is kind of showing you. Our chemically gated channels work. For example, if this was an acetylcholine channel, then the receptor's closed when acetylcholine is not present, but as soon as acetylcholine or whatever neurotransmitter we're talking about binds, it opens the gate, and now we can have the movement of ions. We've got different types of chemically gated channels. This cation channel that we already met. We also have an anion channel, but we'll talk more about those next week. So they open up and they allow their ions to flow down their concentration gradient. Voltage gated channels are closed when we're at rest. So this is showing us a resting membrane. And we've got this potential difference where we're more negative inside than out. But as soon as we have some stimulus like an influx of sodium through our chemically gated channel, and we can reach this positive threshold on the inside, it's going to change the conformation of that channel, and now it will open. And so this is showing us a voltage gated sodium channel. So as we become more positive, it's going to change the conformation, and now sodium can come in. So that's the specific gate. We don't have a picture of a mechanically gated channel, but just be aware they're out there. Alright, so resting membrane potential. This is the difference in charge that we can measure from one side of the cell to another when it's at rest. And nothing's happening. So resting membrane potential is set by our different amounts of sodium, potassium leak channels and our sodium potassium pumps. So we'll talk about that in just a minute. So our resting membrane potential is abbreviated RMP. So this is the difference in charge that we can measure across the neuron when the neuron's at rest. All cells have a resting membrane potential, and their resting membrane potentials are going to be different based on the different numbers of leak channels that they have. So if we look how we measure this, is you stick what's called a voltmeter into a neuron, and you measure what's the difference of charge between the inside and out. And so when we stick a voltmeter into a neuron, we measure the resting membrane potential as negative 70 millivolts. So what is causing this difference in charge? The resting membrane potential is set by this different amount of sodium and potassium ions in the inside and outside of the cell. So you have a lot more sodium outside. You also have a lot more calcium outside. You have a lot more potassium inside. Okay, well, say, well, if potassium has this positive charge, why am I always seeing this negative charge on the inside? It's kind of weird to think about, but remember we said opposite charges are always attracted. So if we look outside the cell, these positive charges are balanced by negative ions that are also hanging out here. We don't talk about the negative ions because they're not important in the signaling. What's important in the signaling is the movement of our positive ions. Just be aware there are negative ions present that are helping to balance it. So like on the outside of the cell it's chloride ion, and the inside of the cell it's phosphate ion, but also you have a lot of negatively charged proteins. So even though what we're watching the movement of is positive charges, our positive charges are balanced by negative charges. And if you measure this resting membrane potential, there's more negative inside the cell than out. And that's really important for setting our resting membrane potential, because if we didn't have this difference, we wouldn't be able to have voltage-gated channels that could change when there's a difference and be able to do signaling. So we have to have this difference maintained so that we can get any kind of signaling at all. And what's going to set the varying levels of resting membrane potential is going to be the differing numbers of these sodium and potassium leak channels that we have. The other thing that helps to maintain this resting membrane potential is the sodium-potassium pump. Because you could just say, well, if we're always leaking, then why don't we reach equilibrium? We don't reach equilibrium because of the sodium-potassium pump. And what it does is it pumps 3 sodium out and 2 potassium in against their concentration gradients. So in our muscle cells, we saw that our resting membrane potential was -90. That's because there are a lot more potassium leak channels in muscle cells than there are in neuron cells. So if we're letting more positive charge leave, then your resting membrane potential is more negative. So that's kind of what's important about our leak channels and our sodium-potassium pump, is they're setting our resting membrane potential. And this video worked for me at home in my lecture, but it does not work on this computer. So just know that you have it if you want to see it. It comes with your Mastering A&P that should have come with your book. You can just watch some videos that kind of express these things to show you the difference in how we're setting up this resting membrane potential. But basically the big players in setting resting membrane potential are these potassium leak channels, the sodium leak channels, and then the sodium-potassium pump. So the most important leak channel as far as setting membrane potential is our potassium leak channel. So we'll say that our potassium leak is the most important for setting our resting membrane potential. And that's because we have more of them, so more positive charge is escaping the cell than positive charge coming into the cell. This is going to allow us to have this negative resting membrane potential. Okay. We also do have some sodium leak channels that are allowing sodium to leak in, but then we're setting up this difference in gradient from one side to the other because of the sodium-potassium pump. So for our potassium leak, we'll say it's most important for setting resting membrane potential. We have more potassium leak channels than sodium leak channels, and they allow potassium out of the cell. So this is positive charge leaving. Our sodium leak channels allow sodium into the cell. And our sodium-potassium pump keeps the concentration of each different. So how do we change our voltage or change our membrane potential so that we can get signaling going? We're going to create what are called other types of potentials. They're potentials because we can measure the difference from one side to the other. And we've got 2 types, what are called graded potentials and action potentials. And they're different. So our graded potentials are these, like that potential when I was showing you the different types of, or the effect of myelin. When we looked at what's happening in the dendrite and we saw that it decayed over time, that's a graded potential. So a graded potential is short-lived, and they travel short distances, and they decay over time. So I'm going to just give you the definition of our two types of graded potentials now. This is where we'll pick up on next Friday, but you should read over it and feel comfortable with it before you come to class next Friday. So the two types of graded potentials that we have are what are called excitatory postsynaptic potentials, or EPSPs, and these are depolarizing, which means not that we go all the way to positive 30, but that we move in the positive direction. So they move potential in the positive direction. And they're abbreviated EPSPs. So our excitatory postsynaptic potentials are little depolarizing events that move the potential in the positive direction so that we can get closer to thresholds. The other type of graded potential that we have is called an inhibitory postsynaptic potential. And these are abbreviated IPSPs. These are what we call hyperpolarizing, which are little events that move the potential in the negative direction. So it'll move you further away from your threshold potential. So again, we'll just define that depolarization is moving away from rest in the positive direction. Repolarization is coming back to resting membrane potential. And hyperpolarization is moving away from rest in the negative direction. These are changing potentials. This is how we change potentials. By depolarizing, that would be changing the potential in the positive direction. Repolarizing is then bringing it back toward our resting membrane potential. And hyperpolarizing is overshooting it so that we move away from our resting membrane potential. So if you think about polarization as being like two opposites. So if our resting membrane potential is negative 70, then we're polarized inside and out. So our cell is sitting here at this resting membrane potential, but we're polarized. We've got opposite charges inside and out. So if we depolarize, then we shrink the difference between inside and out. So we're going to move in the positive direction and make that polarization less. We're depolarizing because we're bringing the poles closer together. When we repolarize, we're coming back to rest and separating our charges again. When we hyperpolarize, we're making this even bigger. We're making the difference even bigger, polarizing them even more. So this figure here is showing you the difference between depolarizing and hyperpolarizing stimuli. So if you're sitting here at rest, and we just have this little graded potential that's excitatory, but not big enough to reach threshold, then sure, you move away from rest in the positive direction. So we're depolarizing a little bit, we're separating the difference, or we're basically making the difference between inside and out a little less, but we haven't hit threshold. So this is what we would call an excitatory postsynaptic potential. It's graded, and it decays over time. Our hyperpolarizing stimulus is going to do the opposite. It's going to take us from our resting membrane potential, and it's going to polarize us even more. It's going to make us a little more negative. So an inhibitory postsynaptic potential does this. It moves us away from rest in the negative direction, but again, it's a graded potential. It decays over time. Why do we come back to rest? Because of our leak channels and our sodium-potassium pump. They set our resting membrane potential. So those things are going to reset us always back to this negative 70 millivolts. So why do we care about all of this? Because this is how we're going to make sure the action potential travels in one direction. So as far as our graded potentials go, these short-lived, short-distance, decaying signals, if you look at a graded potential, if we look at our, like if this is a dendrite and we've stimulated it, then we'll have this, right at the area of stimulus, you have a depolarizing event. So you get like the influx of some sodium ions. So the sodium ions are going to come in here, And the depolarization is going to spread a little bit. So if we had a whole bunch of sodium ions dump in here, those sodium ions are going to be attracted to the negative charge further away from it. But again, we don't have a voltage gated channel here, so over time, those pumps and our sodium potassium pump is going to take back over, pump those things back out, and now your signal has decayed. So our graded potentials are really short-lived, and they can only go a little way away from the area of stimulus. So this is showing if this is our active area where we've stimulated this neuron, or the dendrite, then right at the area of stimulus will have the biggest change in charge. So this would be our depolarizing event. And then those sodium ions can diffuse a bit, but the further you get from the area of stimulus, the more that potential decays. So they can't go very far at all. So, and that's again because our leak channels are and our pump is going to take back over and reset us to the resting membrane potential. So graded potentials are going to be what's happening at the dendrites in the soma. So that we can, basically you can have like, say 100 neurons synapsing onto one neuron. The job then of the soma is to sum up all of those graded potentials and then decide if we're going to shoot an action potential or not. We'll talk more about that next week. The last thing that we're going to talk about today is our action potentials. So when we look at action potentials, we've met them before. We said that they're a wave of depolarization followed by repolarization. What is causing this? So the key players in the action potential is the voltage-gated sodium channel and the voltage-gated potassium channel. And the voltage-gated sodium channel is unique. It has three different states of existence. It can exist in the closed state, where it's like our door's closed and sure, it's locked. Like right now, that door's locked, but the code I put in opens it. The voltage that you put in opens that channel. Then as long as the door's open, sodium can come through, or students can come through. There's a third state for the sodium channel, which is called this inactivation state. And in the inactivation state, we're going to block that, and now you can punch in that code all you want, but it's not going to open. So what I would do to this door to make it like an inactivation gate is I would shut it, I would lock it, I would put up a 2x4, I'd bang it into the wall, and now even if you punch in the code, you can't open that door. And this is going to be important because what it makes sure that happens is that the action potential moves in one direction away from the cell body. The other player that's going to repolarize our cell is the voltage-gated potassium channel. And so it only has two states, open and closed. When it's closed, potassium's hanging out in the cell. When it opens, potassium rushes out of the cell. This is positive charge leaving, so that'll bring us back in the negative direction. So how do our action potentials work? There are four main steps that we can look at. The first step is when the cell's just sitting at rest. And our voltage, all of our voltage-gated channels are shut. So for step one, we can say the cell's at rest. All voltage-gated channels are shut. So our resting membrane potential is being maintained just by these three channels. Our sodium leak channel, our potassium leak channel, and our sodium potassium pump. They're all doing their thing, keeping us at rest. So that's the first step, we just sit at rest. So if we're looking at a graph of the voltage, then we're sitting here at rest and nothing's going on. So resting state, all of our voltage-gated channels are closed. Okay, our second step is going to be depolarization. And this occurs when our voltage-gated sodium channel is open. And I'm going to abbreviate voltage-gated sodium channels VGNC from now on. So the channel is abbreviated this way. So our voltage-gated sodium channel is open, and sodium enters the cell. So What is going to cause these to open is we're going to have to reach our threshold stimulus for our voltage-gated sodium channel, which is negative 55 millivolts. That's going to happen in the axon hillock. The axon hillock is the first place that has voltage-gated channels. Our soma and our dendrites all have chemically gated channels, neurotransmitter gated channels, and they're all generating graded potentials. So if those graded potentials sum up to negative 55, it will open this channel. So we'll say that these voltage-gated sodium channels, open, and this is at a threshold of negative 55 millivolts. And this we got from summing up our graded potentials. We'll talk about summing. This is where we're going to start our lecture next week. But just be aware that we've got these graded potentials, we've got those excitatory postsynaptic potentials that are moving us in the positive direction. And if they sum up and get us from negative 70 to negative 55, we open these voltage-gated sodium channels. So they open, and sodium rushes in till we hit its equilibrium potential, which is positive 30 millivolts. So now I'll say this is our voltage-gated sodium channel, and sodium is going to rush in from negative 55 to positive 30 millivolts. So this is positive charge entering the cell. A lot of sodium is rushing into the cell. It's going to move us away from our resting membrane potential and depolarize us to positive 30 millivolts. So we can see that in this graph here. We hit our threshold of negative 55. That channel opens, sodium rushes in to positive 30 millivolts. What happens at positive 30 millivolts? We begin repolarizing and moving back toward rest. Why? Our sodium channels snap shut, and our voltage-gated potassium channels are going to take over. So our third step is repolarization. So this is going to move us back toward rest. And what happens during repolarization is our voltage-gated sodium channels, they snap shut and they enter their inactivation state. They will not open now, even if you punch in the code. Even if you've got negative 55 millivolts. They will not open right now. So our voltage-gated sodium channels enter their inactivation state. Our voltage-gated potassium channels open. This is going to allow potassium to rush out. So this is positive charge leaving the cell. So now if I've got in red here my voltage-gated potassium channel, potassium rushing out of the cell. So this is positive charge exiting. This is going to repolarize us. I'm not going to put in the voltage yet because there's one more thing that happens with these channels. So repolarization is going to let potassium rush out, bringing us back towards rest. So our sodium channels are going to enter their inactivation state. So now I can't open this gate even with negative 55. And our potassium channels are going to open, potassium is going to start exiting. So as positive flows out, we begin to repolarize. The fourth event is actually hyperpolarization. And what happens in hyperpolarization is that some of our potassium channels stay open past when we hit negative 70. So that more potassium rushes out. And what that's going to do is during that time, our voltage-gated sodium channels are going to reset to closed. And what that's going to do is going to allow for what's called like this relative refractory period, where now the gate is closed. So I could open it, but I have to really, really want to open it because now I've got 10 more millivolts to overcome than before. So what this is going to do is it's going to reset our sodium channel. It's possible to open it now, it's just a little harder because we've got a bigger voltage difference to overcome. So our hyperpolarization is when some potassium channels stay open to about negative 80 millivolts. And what is happening during this time is our voltage-gated sodium channels reset to closed. And so now they're reset to closed. So if we want to open them, we can, but now we've got to go, instead of from -70 to -55, we've got to go from -80 to -55. So you have to really want to open it. But it's possible. It's just a little harder. And what this is going to do, this whole chain of events, especially this sodium entering its inactivation state, is it makes sure that the action potential travels in one direction away from the soma. So if you think about it, our plasma membrane has voltage-gated sodium channels down the whole thing. So if we depolarize right here, I wish my pointer was working. If we depolarize right here, then what's going to happen is we'll open up these next voltage-gated sodium channels. So then these ones are open, but what's to prevent us from going back and opening the ones that were just open? If -55 is what does it, and we're bringing all this positive charge, the inactivation state does it. So what's happening, if you look as you move down your neuron, that inactivation gate is making sure that even though we've got positive charge coming in here, it can't go back and open up these voltage-gated channels down here because they're in their inactivation state. And so that keeps the action potential moving in one way away from the soma. So they go in one direction, and once they start, they're self-propagating. Why are they self-propagating? Because as soon as we depolarize this voltage-gated sodium channel, sodium is going to come in and cause us to reach threshold at the next one. And that's going to cause us to reach threshold at the next one, which causes us to reach threshold at the next one. So as long as we keep bringing in that positive charge, it will self-propagate because of those voltage-gated channels. And it's prevented from going back because of the inactivation state. Another video that I recommend watching that comes with your book, I think we can actually get this one on YouTube. So we'll look at this in just a second. And what's important about it, or what's important about the refractory period, is that if we didn't have that absolute refractory period, then it wouldn't matter if we had a stronger stimulus. Like, we would only be able to generate this number of action potentials. But if we've got a really strong stimulus, you might want to be able to increase the velocity of your action potentials or make them happen faster. So now this is what it's showing here. If we overcome that voltage difference, so if you think about it, at first from -70 to -55 is 15 millivolts that we've got to overcome. So we've got all these excitatory postsynaptic potentials adding up to 15 millivolts. Well, if we go to hyperpolarize from -80 to -55 is now a 25 millivolt difference. So now I've got to have a lot of excitatory postsynaptic potentials hitting that neuron. But if I do, then I should still be able to fire an action potential.